Hello and welcome. Hello and welcome to the week 12 live session. This will be the last live session on the course Experimental Biochemistry. Okay, so I am Srijan Sen and I will be taking your live session on the course Experimental Biochemistry, which is being taught by Professor Shagata Das Gupta and Professor Shomode of IIT Kharagpur. So I'm sharing my slides with you all and you confirm me once it's visible. So first I will be discussing the lecture slides in brief, and then we will be talking about the assignments. Okay, right. So today's class will mostly cover up the blotting techniques. Okay, so we will talk about the different techniques of blotting and why we need to do blotting and what are the advantages of doing blotting, what are the different types of blotting methods we know or we need to know. So what is blotting? So blots are techniques for transferring DNA, RNA and proteins onto a carrier so that they can be separated, and often follows the use of a gel electrophoresis. So the southern blot is used for transferring DNA, the northern blot for RNA, and western blot for proteins. So there are different blotting techniques and types of blotting that we can use. The first one is the eastern blot. We have western blot, southern blot, and the normal blot. So like a compass, you can actually, in a compass, you have the north, south, east, and west. So for blots also, we have the different types of blots, like the eastern blot for detecting protein translational modifications. You have the western blot for protein detection. For detecting DNA, we have the southern blot, and for detecting RNA, we have northern blot. So southern blot is the blotting that was named after the scientist E.M. Southern in 1975. And after that, the concept of northern, eastern, and western, on the basis of how we divide a compass, on the basis of that, we have um, moved ahead with the other different types of blotting. So, like I said, the southern blot is used to detect the presence of a particular piece of DNA in a sample. So the DNA detected can be a single gene or it can be a part of the larger piece of DNA, such as a viral genome. It combines agarose gel electrophoresis for size separation of DNA with methods to transfer the size separated DNA to a filter membrane for probe hybridization. The key to this method is hybridization. So what is hybridization? So hybridization is a process of forming a double-stranded DNA molecule between a single-stranded DNA probe and a single-stranded target patient DNA. So the principle is that the mixture of molecules is separated, the molecules are immobilized on a matrix. The probe is then attached to the matrix to bind to the molecules. Any unbound probes are then removed. The place where the probes are connected corresponds to the location of the immobilized target molecules. Okay, so this is the apparatus behind the southern blotting technique. So you see you have a weight of less than 0.75 kg, then you have the glass plate followed by paper tissues, three sheets of filter paper, you have the membrane, gel, and then you have the plastic tray for the capillary plotting apparatus. So the steps in southern blotting, you see you digest the DNA with an appropriate restriction enzyme. The complex mixture of fragments is then subjected to gel electrophoresis to separate the fragments according to the size. The restriction fragments present in the gel are denatured with alkali and transferred onto a nitrocellulose filter or nylon membrane by blotting. This procedure preserves the distribution of the fragments in the gel, creating a replica of the gel on the filter. Then what you do, the filter is then incubated with the hybridization conditions with radio labeled DNA that's specific for the particular DNA sequence. Then the probe hybridizes to the complementary DNA restriction fragment. Whatever the excess probe is there is washed away, and the probe bound to the filter is detected by autoradiography, which reveals the DNA fragment to which the probe is hybridized. So in summary, what we have done, it's in 8 steps. First is extraction and the purification of DNA from the cells, restriction of the DNA with the different restriction enzymes and all. Then we carry out the electrophoresis, agarose gel electrophoresis, denaturation of the DNA, then the transfer process, blocking of the excess DNA, washing of the unbound probe, and finally detection technique. So you see you digest the DNA with appropriate restriction enzymes. The complex mixture of fragments is subjected to gel electrophoresis to separate the fragments. Then you transfer it using onto the nitrocellulose membrane by blotting. You have then the process of hybridization for binding of the specific radiolabel probe to your specific DNA of interest, and the excess probe is washed away and the probe bound to the filter is detected by autoradiography. So the applications include southern blots are used in gene discovery, mapping, evolution and developmental studies, diagnostics and forensics, identification of the transferred genes in transgenic individuals. Okay, now we have northern blotting. So what is this northern blotting? For the transfer of RNA, okay, it's very similar to that we did for DNA. So it was developed by James Alvin and George Stark of Stanford University and was named by the analogy to southern blotting. So what do we have over here? So isolation of the nucleic acid, that's RNA over here, from the biological samples. Then RNA is more susceptible to degradation of DNA, hence handling RNA should be done with a lot more precautions. Then what we have, the sample is loaded on the gel and the RNA samples are separated according to their size on an agarose gel. The resulting gel, following after the electrophoresis run, then the gel is blotted onto a nylon membrane or a nitrocellulose filter paper or diazobenzylomethyl papers by creating the sandwich arrangement like we did. That's a transfer. The membrane is then placed in a dish containing the hybridization buffer with the labeled probe that can be that's radio labeled, and thus it will hybridize to the RNA on the blot that corresponds to the sequence of interest. The membrane is washed to remove the unbound probe, and then you observe it under the autoradiogram or via some chemiluminescence if you have used a chemically labeled probe. In both the cases, this results in the formation of a dark band on the X-ray film. Now, the expression patterns of the sequence of interest in the different samples can be compared. So you see, you have similar uh, diagrams like over here. You have the RNA is isolated from the biological samples. Then you run it on the gel. You see, you have 32p labeled size markers. Then they are separated on the agarose gel according to their size. You transfer it on an amino benzyl benzo hydroxy methyl filter paper by creating the sandwich arrangement. It's then placed in a dish containing the hybridization buffer with the labeled probe. Then you comes the detection technique. So what are the applications? It's that a standard for the study of gene expression at an mRNA level can be understood. You can detect the mRNA transcript size, study RNA degradation, RNA splicing, RNA half-life. Then it's often used to confirm and check the transgenic knockout mice animals. Now there are a few disadvantages of northern blotting. The first one is that you're handling RNA, so you have to be a lot more cautious whenever you're doing anything. So the standard northern blot method is relatively less sensitive. Detection with multiple probes is a problem. Now, if RNA samples are even slightly degraded by RNases, that's often happens. So the quality of the data and the quantification of expression is affected. Another widely used blotting technique is the Western blot that was developed in 1981, which is an immunoblotting technique which relies on the specificity of binding between a protein of interest and a probe to allow the detection of the protein of interest in a mixture of many other similar molecules. The SDS page technique is a prerequisite for Western blotting. So the steps in Western blot is that a protein sample is subjected to electrophoresis on an SDS page. Electroblotting transfers the separated proteins from the gel to the surface of the nitrocellulose membrane. The blot is then incubated with a generic protein like skimmed milk proteins or BSA, which binds to any of the remaining sticky places on the nitrocellulose. Now, any antibody that is specific for the protein of interest, that's the primary antibody, binds to the nitrocellulose sheet and reacts with the antigen. On the band containing the protein of interest binds the antibody, forming a layer of antibody molecules. Okay, so then only the band containing the protein of interest binds to the antibody, forming a layer. So after washing for the removal of non-specifically bound antibody, secondary antibody is added, which specifically recognizes the Fc domain of the primary antibody and binds to it. Now, antibody 2 is radioactively labeled or is covalently linked to the reporter enzyme, which allows to visualize the protein antibody 1 antibody, the primary antibody, and the secondary antibody complex. So you see, you do the lysis, then separation of the protein. So first is preparation of samples, protein extraction, then you do SDS page, then transfer of the onto the PVDF or the nitrocellulose membrane, whatever you're using, then blocking the membrane using some kind of proteins, like you have the BSA, some blocking agent like the milk solution or something. And then you have the detection. So the application is that the confirmatory HIV test, it's for the definitive test for bovine spongiform encephalopathy, the BSC, then some forms of Lyme disease testing employ Western blotting. So these are the different techniques that the three mostly the widely used blotting techniques. It's a very short one, so yes, we will talk about the practice problems now. Okay, so let us talk about the questions. So what is the first step of Western blotting? To run an agarose gel, to run a SDS page, to transfer the proteins in a membrane, to add primary antibody to the sample. So the first thing that happens is that you have to run a SDS page, and then you go for the transfer, and then the primary antibody is added, which is incubated for around 16 hours. Now Western blotting is performed to detect is it specific DNA, RNA, specific protein, or DNA protein complex. So what is the specific DNA? For specific DNA is southern blotting. Northern blotting is for detecting of RNA. Western blotting we do for specific proteins, and the DNA protein complex is known as IMSA or electromobility shift assay. That's IMSA. Okay. Western blot technique is also known as immunoblotting, immunoprecipitation, southern blotting, or hybridization. So what is it? Yes, immunoblotting is another name that we give because there is the primary antibody that is binding to the antigen, which is our protein of interest. So we call it the immunoblotting. The proteins in a sample are separated using SDS page, Western blot, protein purification, or immunoblot. So how do we separate the proteins in a sample? We have to run an SDS page. The next one is the for DNA fingerprinting. Which of the following technique is used? So for DNA fingerprinting, what you use? We use southern blotting. Now in blotting techniques, a probe is, what is a probe? A specific protein to detect DNA, a short piece of labeled double-stranded DNA which is complementary to a specific DNA, a short piece of labeled single-stranded DNA or RNA which is complementary to the specific DNA or RNA, or a specific protein to detect RNA. So a specific protein to detect DNA, that is not right. A probe, a short piece of labeled double-stranded DNA which is complementary to the specific DNA, that is also not right. Rather, a short piece of labeled single-stranded DNA or RNA which is complementary to the specific in RNA, because it also has to bind to the DNA such that at the end of the day we can do the detection and see it under a by autoradiography. So which of the following technique is suitable for detecting mRNA expression in cells? So we know that for RNA we do northern blotting. So from here you are going to get very direct questions, nothing very tough. The non-specific sites on the PVDF membrane can be blocked by twin-20, non-fat dry milk, SDS, sodium chloride. So for as a blocking agent, we need to use some kind of proteins, that is our milk protein. So we can use the non-fat dry milk. Twin-20, SDS page are simple detergents that we use during the washing. We do not use SDS, but we use Twin-20 for washing of your So we use for washing of the membrane during the different washing times for the removal of the unbound antibodies and all. Okay. Why the probe is labeled? So why does the probe have to be labeled? To improve binding capability. That is not right, because there has to be a specific binding. So depending on the specificity, the binding will happen. To improve location identification, that is absolutely right, because you need to identify the position of where your DNA is, to improve visibility and stability. So to improve uh, visibility, that is also correct. What is the major force behind protein and or nucleic acid to go out from the gel onto the membrane? Electric current, pH gradient, voltage, or temperature? So the major force is that you do not use a pH gradient, because then it would affect your DNA or RNA or whatever the proteins you're using. It's basically the electric current and the voltage that you are applying. Okay. Why labeled antibodies are used? So we use labeled antibodies. Why? To detect the presence of particular DNA molecule in northern blotting, presence of particular RNA molecule in southern blotting, presence of particular protein molecule in western blotting, or to presence of DNA molecule in southern blotting. So why do we use it? So to detect the presence of DNA in northern blotting, that's wrong, because we do northern blotting for RNA and southern blotting for DNA. So A and B is wrong. Presence of protein molecules in western blotting, this is correct. Or to detect DNA molecules in southern blotting, but that is not correct, because we use probes, not antibodies. Which of the following is or are used to detect the protein in western blotting? So antigen, antibody, DNA or RNA probe. So antibody is the correct answer. Which of the following membranes is best if you want to strip an already used membrane for Western blotting? So which of, so in other way, which of the membrane can be used for um, which of the membrane can be reused? Is it the nylon membrane, PVDF, nitrocellulose or paste? Yes, it is the PVDF membrane that you can use, not the nitrocellulose one or the nylon membrane. If you forget washing the membrane after incubation with primary and secondary antibody, what will happen? Background noise will be high, whole blot will be dark, no specific bands will be observed, no change in blot will occur. So what are the problems if you forget to wash after the primary or the secondary antibody? So first is that there will be lot of unbound primary and secondary antibodies that will be there. So the background noise will be very high, because of which the whole blot will be dark, and finally you will observe you cannot identify the bands. The last question, why are ghost bands appearing in the blots? So what is the probable reason? Too much protein has been loaded, blocking was not done, incubation with primary antibody was too long, too much of substrate for secondary antibody was added. So ghost bands appear because there might be that too much of protein has been loaded, because of which you see an elongation in the band length, and because of which the binding is more, and your the band appears at very random positions. So those are the ghost bands. The other reason is that you have incubated with primary antibody for a long time, because of which you see ghost bands. That's because of the non-specific binding. Okay, so these are the two probable reasons. Okay, so that's all then. Thank you. So this is my last class. Hope you give a very good exam, and if you have any problems, you can just mail me, and the NPTEL team will be sharing the slides with you all. And the video lectures are uploaded in YouTube. You can check them anytime you feel you are facing a difficulty, and give your exam fantastically and do good. So best of luck to you all, and yeah. So thank you all for joining all my classes and attending it. And so that's all from my part then. Okay, so best wishes are with you guys. Thank you.